design and the development of nanoparticles for personalized medicine. So what is nanomedicine? As you know, nanomedicine is the application of nanotechnology to achieve breakthroughs in healthcare by exploiting the improved physical, chemical, and biological properties that materials have at the nanoscale, which results in a better targeting and bioavailability of therapeutics and new modes of therapeutic action. We know that these improved characteristics depend on several factors, mainly on the nanometric scale dimensions, which enable the fine-tuning of nanoparticle surface due to the enormous surface to volume ratio, and the possibility, for instance, to encapsulate and protect labile compounds such as genetic material or proteins or peptides, and protecting them from the degradation by the enzyme processes such as proteases or plasma nucleases. And still the possibility to encapsulate chemically different molecules, and in particular to improve the pharmacokinetic properties of those drugs that have a very low water solubility. And last but not least, a specific organ targeting through the selective delivery of therapeutics to the site of the disease while sparing off-target effects. However, from the site of injection to the site of the disease, several sequential barriers impede and hinder the therapeutic efficacy of those nanoparticles. And in particular, we have seen so far that they have limited the potential clinical translation of currently used nanoparticles. And we mainly focused on the role of the mononuclear phagocyte system, and in particular on the macrophages, since those are the sentinels in our body, both circulating or located into the main filtering organs such as liver and spleen, that are able to recognize a non-self material such as a nanoparticle and rapidly clear it, thus reducing the half-life. On the same At the same time, the macrophages represent an ideal carrier within the body, since they can freely circulate in the bloodstream, and in particular, they can sense whether something is going wrong and infiltrate any kind of tissue following to an inflammatory process. And we know that inflammation is the first line of defense response to an injury or an infection, but it's also the background of several pathologies, such as cancer, cardiovascular diseases, Alzheimer, or autoimmune diseases. So we thought at this relationship between the nanoparticle and the macrophage as a relationship that in nature there is between a prey and its predator. And we know that in nature the prey to escape from its predator uses the biomimicry, which is a technique to look like the predator itself. So we thought to make nanoparticles that could look like macrophages in order to exploit these peculiar properties of macrophages or leukocytes in general to escape the immune system and at the same time target the inflamed vasculature and the inflamed tissues. So we developed leukosomes by combining the membrane proteins of leukocytes with synthetic biocompatible lipids such as cholesterol and choline-based phospholipids in a liposome-like carrier where membrane proteins are embedded within the lipid bilayer. And our goal was to create a particle that could camouflage within the bloodstream, that could escape the surveillance of the immune system and reaches its target undiscovered. We have characterized these biomimetic nanoparticles both from a physicochemical and biological standpoint. Here you can see the physicochemical characterization, which revealed a size of 120 nanometers, a homogeneous formulation in size, as you can see from the PDI polydispersity index value and the cryo-EM analysis, while atomic force microscopy analysis and FTIR revealed a rough surface for those particles due to the presence of membrane proteins. The with-germine agglutinin assay indicated that the proteins on leukosome surface are glycosylated, which means that they are in their post-transcriptional active status and giving insight into the correct orientation inside the lipid bilayer. Flow cytometry analysis identified the markers that we were actually looking for, and in particular those receptors on leukocyte surface, such as LFA1, MAC1, CD18, and PSGL1, that are able to recognize and bind to the cell adhesion molecules that are overexpressed by the endothelial cells in case of activation or inflammation. In addition, we could identify CD45 and CD47, which are recognized as self-tolerance and do not eat me signals. The biological evaluation of these leukosomes allowed us to study their behavior once injected in vivo. And as you can see, compared to conventional liposomes, leukosomes showed a decreased accumulation in the liver and spleen and a prolonged circulation when in the bloodstream. We then investigated the targeting properties, and we used an in vivo model of localized inflammation. We subcutaneously injected lipopolysaccharide into the right ears of the mice, keeping the contralateral ears as control. And as you can see, both at 1 and 24 hours after injection, we could observe an increased accumulation of leukosomes in the inflamed ear. Here you can see leukosomes fluorescently labeled in red, while vessels were stained with high molecular weight dextran using intravital microscopy analysis. And we dissected the mechanism of these targeting properties by using blocking antibodies against LFA1 and CD45. And in particular, we observed that by blocking those receptors on leukosome surface, we abolished their targeting properties. We were handling biological materials, so cell membranes. We wanted to make sure that we were not triggering any immunogenic response. So we have treated biopsy mice weekly for 4 weeks. Then we incubated the serum withdrawn from these mice with leukosomes and control liposomes, and we did not observe any elevation of autologous antibody titer compared to the control. And in particular, less than 0.3 and 3% of leukosomes were labeled by host serum and secondary antibodies containing particularly anti-IgG and IgM in levels similar to control liposomes, thus indicating that leukosomes do not initiate any adaptive immune response and antibody production against membrane antigens related to the particles. As I mentioned before, our main goal was to use the leukocyte arsenal to confer nanoparticles active targeting properties in the context of a drug delivery carrier. So we have encapsulated in leukosomes a potent corticosteroid, dexamethasone, and we tested the in vivo anti-inflammatory activity in the localized inflammation model that I showed before. You can see here in those immunofluorescent stainings where nuclei are in DAPI while LY6G neutrophils are in red, the group treated with dexamethasone-loaded leukosomes, which is the group number 6, showed a reduction of the neutrophil infiltration, which means a reduction of the inflammatory processes. But what intrigued us in this experiment was the behavior of the mice of group 5, which are the mice treated with empty leukosomes, since we could observe in this group a reduction of the inflammation. But I will come back to this later. So we move to test our leukosomes on other models that share an inflammatory background, and among these, cancer has been often referred as a wound that never heals. In particular, we tested doxorubicin loaded leukosomes in 2 models of breast cancer and melanoma model. We could observe a accumulation greater than control liposomes in those 2 models. And in vivo, we observed a reduction, a delay of the tumor growth, and at the same time, a prolongation of the mice survival. Here, in another cancer model, a medullary thyroid carcinoma model, in collaboration with Dr. Angela Carter and Professor Bibbs from Alabama University, we evaluated the enhanced delivery of anti-CDK5 therapy using leukosomes, and we demonstrated a time-dependent spread from the vasculature into surrounding tumor tissues of these biomimetic nanoparticles, as you can observe in A, and increased tumor localization compared to control liposomes in B, a 6-fold plasmatic stabilization of encapsulated drug compared to the free one in C, and delivering only 5 milligrams kg biweekly of Indo-A encapsulated in leukosomes, we could observe the same effects as delivering 20 milligrams kg biweekly of free drug. But we protected animals against hepatic and renal toxicity, as you can observe in E and F. We then tested another inflammatory model, per excellence, an advanced atherosclerosis model. In vivo targeting experiments revealed an increased accumulation of leukosomes to the site of the atherosclerotic plaque compared to liposomes, and the loading of rapamycin within leukosomes could increase the therapeutic anti-inflammatory and anti-proliferatory effects on the vessel wall by obtaining a decreased percentage of proliferating macrophages, a decreased enzymatic activity in aorta extracts after 7 days of treatment, and reduced rapamycin adverse systemic events. I mentioned before that we were intrigued by the activity of empty leukosomes, since we found that those nanoparticles could have an intrinsic anti-inflammatory properties. So we decided to test those properties in a severe inflammatory model, which is the endotoxicosis model of sepsis, obtained through the intraperitoneal injection of lipopolysaccharide. This is a very nasty model. Mice die between 18 and 24 hours if untreated, and mainly because of respiratory distress. We have injected once leukosomes free so empty for this experiment, and compared their activity to the one of the free dexamethasone, the potent corticosteroid, which is clinically used for these treatments, and empty liposomes. And first of all, we could observe an increased accumulation at both 1 and 6 hours after injection at the lung parenchyma. And in particular, looking at the survival curve, the 66% of mice treated with empty leukosomes, the line in red, could survive, compared to the 50% of mice treated with the free drug, while mice treated with liposomes died over the 48 hours. And these effects in vivo were associated to an improvement of the lung histological score. You can see in these videos how mice look like after the various treatments. Liposomes and dexamethasone treated mice were pretty hunched and lethargic, while you can see the leukosomes treated mice were at 24 hours already moving around and recovering very quickly from the endotoxicosis model. We then studied in vitro a possible mechanism of action of these nanoparticles, and we hypothesized that the intrinsic anti-inflammatory activity of leukosomes is mediated by their interaction with J774 macrophages, thus behaving like synthetic exosomes in order to communicate with macrophages and induce them to reduce the expression of pro-inflammatory chemokines and cytokines. At this point, what reviewers were always asking to us was how we envision to clinically translate the manufacturing of these nanoparticles. And we know that large-scale nanoparticle manufacturing actually represents one of the main limits to their clinical translation. Manufacturing protocols for biomedical application require that the final product is standardized, batch-to-batch consistent, scalable, GMP compliant. And in the past years, we observed that microfluidics provided a versatile tool for the well-controlled synthesis of nanoparticles in order to accelerate the transition to the clinic. And in particular, by playing with the operating parameters like the flow rate ratio, the total flow rate, and the temperature, we could fine-tune the nanoparticles parameters, final size distribution, loading capacity, bench-to-bench reproducibility, and protein incorporation into the lipid bilayer. At this point, we have physicochemically characterized these microfluidic assembled leukosomes and compared to the previous one, which were assembled using the thin layer evaporation method. And what we found out was that using the microfluidic approach, we could first of all increase the protein to lipid ratio from 1 to 300 to 1 to 50. And compared to the previous thin layer evaporation method, the microfluidic procedure allowed for a 14-fold increase of protein concentration on the surface of leukosomes and a 1.6-fold increase in vivo accumulation of nanoassembled leukosomes at the site of inflammation. To conclude, we have shown that the bio-inspired nanomedicine represents a novel way to conceive drug delivery. Our biomimetic approach allowed for the transfer of leukocyte membrane proteins to the surface of a synthetic carrier, a liposome-like carrier, in their correct orientation and in their active post-transcriptional status. Leukocyte proteins bestow vesicles with biological properties and intrinsic anti-inflammatory activity in several inflammatory-based models, and microfluidics offered a universal and versatile tool to formulate biomimetic nanoparticles in a scalable, reproducible, standardizable, and GMP transferable fashion. Those are all the people involved in my training as a scientist, but also from a personal standpoint. And I would like to thank them all. Thank you for your attention. And I leave you with this take-home message. And thank you very much.